We're now going to explore organic compounds and how they are made and broken down. So making them is referred to as synthesis, and breaking them down is a process called hydrolysis. So what are organic molecules? They're going to be molecules that contain compound carbon. And we have exceptions. Carbon dioxide and carbon monoxide are considered inorganic. Carbon itself is electroneutral, which means it does not form ions, so it doesn't gain or lose electrons. Carbon can form 4 covalent bonds. That's something important to remember. So anytime I draw carbon, I'm going to draw 4 dashes around it, indicating that it can form 4 covalent bonds. And carbon is unique to living systems. The fact that carbon can form 4 covalent bonds even with itself and many other elements gives us many combinations of compounds that can be produced. The major organic compounds found in our bodies include carbohydrates, lipids, proteins, and nucleic acids. Many of these compounds are polymers, and a polymer, poly means many, is going to be a chain of smaller subunits called monomers, which are the building blocks. So these would be several repeating units of the same type of molecule. For example, complex carbohydrates are made of simple sugars. Those would be the monomers. They're going to be synthesized by a dehydration synthesis, which means we have to remove water. And then we're going to break them down by hydrolysis, which is the opposite. We're going to add water. So hydro splitting is the literal translation for hydro, and lysis is to split. So here's an example of a dehydration synthesis. We have 2 monomers, monomer 1 and monomer 2. One has an OH group called a hydroxyl, the other one has a hydroxyl as well. And if we remove the hydroxyl from one and a hydrogen from another and put those together, that makes the water molecule that's going to be leaving. And the result then is a dimer, or a molecule made up of 2, a larger molecule made up of the 2 molecules that were going through the dehydration synthesis reaction. So there's the new covalent bonds being formed in that process. Hydrolysis is the opposite. If we start with a larger molecule and then we bring water in here and add the water will help to break apart the covalent bonds, and one of the monomers receives OH and the other one receives the hydrogen. Here's an example here in the production of table sugar called sucrose. Sucrose is made of 2 simple sugars, one called glucose, and the other fructose. Both of them are 6-carbon sugars, but you can see here the OH on one sugar, the glucose, and the H from fructose are going to be removed in the equivalent of water and forming a covalent bond here. And this gives us sucrose. And we can do the opposite, so that was a dehydration synthesis. If we wanted to split them up, we would add water. So we'd add the water, and the water would go back and help split the molecules, one molecule receiving OH and the other one receiving the H or hydrogen. So we then have separate glucose and fructose. So that would be an example of specific example of formation of a larger molecule by dehydration and the splitting using water. Now we're going to look at carbohydrates. Carbohydrates include simple sugars and starches. They contain carbon, hydrogen, oxygen, and the hydrogen and oxygen are usually in a ratio of 2 to 1. There are 3 classes. There are monosaccharides, and saccharide means sugar. Okay, so this is the smallest unit of a carbohydrate. Then we have disaccharides, which is 2 sugars, and we just saw sucrose as an example of a disaccharide. And then we have polysaccharides, which means many sugars. These are to be polymers that are made up of the monomers called monosaccharides. So monosaccharides are going to be simple sugars containing 3 to 7 carbon atoms. And there's a general formula for the carbohydrates. The N outside right here refers to how many carbons there are. So if there's 3 carbons, then we would have 3 carbons, and then the 3 times the 2 is now 6, and then 3 oxygen. So it would be basically, it's always a a 1 to 2 to 1 ratio, and it depends on how many carbons there are. So if it's 6 there, if we have 6 carbons, then it would be C6H12, because we have to multiply the 6 by the 2, and then O6. If it was a pentose, so this is actually a hexose, hexa meaning 6, then it would be 5 carbons, so then that would be C5H10O5. Okay. So the monomers here for monosaccharides, the monosaccharides are the monomers of carbohydrates. Important monosaccharides include pentose sugars or pentoses like ribose and deoxyribose, which are found in RNA for ribose and DNA for deoxyribose. So that's where the D comes from, deoxyribose, deoxyribonucleic acid, and then ribose for ribonucleic acid. And important hexose is sugar, is the sugar glucose, which is what is measured when we measure blood sugar. Here are some of the monosaccharides. We have, you're not responsible for memorizing the structural formula, but this just shows that there are differences in the way the atoms are arranged, or groups of atoms. We have glucose here, then there's fructose. We have also have galactose. And so there's just differences basically in the way the atoms are arranged together. So they're isomers of each other. And then here we have the pentoses, deoxyribose found in DNA, and ribose found in RNA. Disaccharides, di means 2, so these are double sugars. These are going to be too large to pass through cell membranes, and they include sucrose, maltose, and lactose. So if we're going to ingest these, we need special enzymes to hydrolyze them, use water to split them into their basic sugars. If you're lactose intolerant, that's going to be because you don't have an enzyme to break down lactose into its simpler sugars. So then that sugar doesn't get absorbed in your small intestine and travels to your large intestine. There's plenty of bacteria in there just waiting to break down the lactose, and then the products of that are what may make you sick with cramping and gas and that kind of stuff. And a loose stool. So that's the explanation for lactose intolerance. The disaccharides are going to be formed by dehydration synthesis from 2 monosaccharides. For example, we saw earlier a picture of glucose and fructose removing the water, giving us the sucrose, and there's that water that was removed by joining the 2. Here are some of the disaccharides pictured here. Sucrose is a disaccharide of the monosaccharides glucose and fructose. Maltose is made of 2 glucose molecules, and lactose is made of galactose and glucose. So what we have there for lactose intolerance here would be an inability to break down that bond there, and so we can't break it down to the simpler sugars. And so that's when you get sick, or can get sick, or have stomach issues. Then we have polysaccharides. Polysaccharides are polymers of monosaccharides. They are also formed by dehydration synthesis of many monomers. Important polysaccharides include starch, which are found in plants. Okay? And starch is just made of glucose molecules. Animals, on the other hand, store their glucose, same kind of monosaccharide, in the form of a polysaccharide called glycogen. And all it is basically a difference in the way the glucose molecules are bonded together. Glycogen is going to have more branching. And so it's how glycogen is how it's stored in animals. Neither one is very soluble in water. So they do have to be broken down so they can be moved around within cells and within your bloodstream, those fluids are water-based. Okay? So here's a picture of polysaccharides made of many monosaccharides, and this is glycogen. You can see the branching. Each little hexagon on there represents a glucose molecule, and you can see here how the glucose molecules are bound together, and a branching. So right here we see one of the branching chains there. Just to be sure that sometimes when you see a molecule structure and you don't see any of the atoms on there, then everywhere there's a bend, it's understood that in that bend is a carbon. Okay? And then if we don't have our complete octet, you don't see the other elements, then you might assume that around each of the carbons it's filled with hydrogens, unless you draw something otherwise. For example, right up in here in this corner, instead of leaving it just a bend in the lines there, you see an O there indicating it's not a carbon, it's an oxygen.